Can I ask here?
Yeah, so
Okay.
I'm not sure. Everyone has the link and they know about the class date and time and how to access it. As to why they're not here, I am unaware.
Okay, thanks. I was just wondering.
Okay. Okay, and the other thing that I wanted to discuss is I've already emailed you the files and the presentation for today's lecture. So I'm hoping that every one of you would have received it. If you haven't, so please let me know. Okay, great, Bharat, thank you. Okay, so hoping that there are no questions to be asked, so we'll start with today's lecture. Julia, all yours.
Okay, well, thank you, and thank you for listening. So today we're going to be actually doing something very similar to what we have been doing both on Tuesday and on Friday, but we will complicate things a little bit. And by complicating, I mean complicated data set that sometimes we have to deal with anyways. So we're going to talk about discriminating between toxic and non-toxic molecules based on their physicochemical descriptors. Now, the goal will be the same, basically the same as we had before, to find descriptors or that discriminate between toxic and non-toxic molecules. Where did we get this data of molecules? The data was obtained from the two data sets, these ones that you have seen before. No, maybe not. The two data set, one is a data set of toxic molecules, and the other one is a database of metabolites. From there, we have extracted 12,000 toxic and non-toxic molecules that are characterized by by some words that are called descriptors. We chose the descriptors in the following manner. We chose only descriptors that were in both sets, in both the toxins and the metabolites, so that the discriminants won't be very clear. And each descriptor has a value of -1 if this descriptor is present in the molecule, or 1 if it's not. Let me share with you the table, how it actually looks. So this is the original table. Here you have an ID of a molecule. And here you have descriptors. For example, it can be an anti-infective agent or some chemical property like guanol. And we have assigned, just if the molecule in the database has this descriptor, we assign a 1. And if it doesn't have, we assign -1. So what I mean by problems in our data set is the following. First, our data is not normally distributed. Obviously, it is binary data, so there's no normality. And for linear discriminant analysis, it assumes normality. And the second problem that we ran into was that some of the columns, so some of the descriptors, were very, very correlated. And again, this can cause, now, random forest can deal with non-normal data. It usually deals fine. But correlated data set, correlated columns of the table can be a big problem for both linear discriminant analysis and nonlinear discriminants like random forest or decision trees. So this was the challenge. This is a challenge that we think you can come into also in your data if, for example, you're looking for some descriptors of a disease or some other descriptors that are, again, that can have numeric values and are not normally distributed. So what can be done to overcome this? Well, instead of dealing the original table, we can move to the PC space. And moving to the PC space actually takes care of both of these problems. Usually, PC coordinates are normally distributed, and even if not, remember that random forest deals fine with non-normal distribution. And of course, PC vectors are orthogonal, meaning that they are uncorrelated. But there is a little trick, and the trick is the following. As you remember, hopefully, each PC is dependent on all features. So if we move to the PC space, we will still be dealing with all of our descriptors, which is not good because we want to choose a very small amount of descriptors that can discriminate between the molecules. So what we will do, we will we will erase the descriptors that don't have enough contribution to the PC. And that will make our table smaller. So I hope you're following. Please let me know if this is getting too hard or hard to follow or unclear. I can stop and try again. Okay, so what we will do, this is what we will do. This is a PC space with all descriptors. This is a PC space of descriptors. But we don't want descriptors in the PC space. We want, in this PC space of descriptors, we want to see molecules. But before we do that, we want to erase some of the descriptors from this space. So what we will do, we will look at the distribution of all the coefficients of PC coefficients. We will find the average of the coefficient, and we will erase these tails. So if the descriptor has a coefficient that falls in this tail or in this tail, we will make its contribution or its coefficient equal to 0. This way, when we will finally find the PCs that discriminate between toxic and non-toxic molecules, because that's what we will do. We will work, the discriminant analysis will work with the PCs and not with the descriptors, straight with the descriptors. Some of our descriptors will have zeros on this PC, zero input on this PC. And then we will just not look at them. We will look only at the descriptors that have non-zero impact on the PC or non-zero coefficients. This is the little trick. Then we will move back to the molecule table and to the descriptor table, and we will try to understand which descriptors are actually separating the toxic and non-toxic molecules. Okay, so this is a little outline of what we're going to do. So to help you a little bit, I hope it does help. We have a table of molecules, and we have a problem with this table. And the problem is that the features are dependent and are not normally distributed. So we will move to the PC space where we overcome both the non-normality and the dependence problems. We will perform supervised discriminant analysis and identify the PCs that discriminate between toxic and non-toxic molecules. And then we will attempt to move back from the PCs to the descriptors for some biological interpretation. Can we actually find a way how to discriminate between toxic molecules and non-toxic molecules based on chemical physical descriptors. So first of all, let us open the files. Let me open the files to show you how the file actually looks. This is the original table. We have names of molecules here, and actually we know who is who. If it came from T3D database, it is a toxic molecule. If it has a name of this sort, starting with HMDB, this is a metabolite. So we assume it is non-toxic. From the databases, we selected 140 chemical physical properties, and we put some value, -1 if the molecule doesn't have this descriptor, and +1 if the molecule does have the descriptor. Now, what we're going to do with this table is we're going to move to a new space, to the PC space. As you understand, if I do PCA on this table, I will get a table of how my descriptors are assembled, are distributed in the PC space. So I will have the coefficient, PC coefficients for the descriptors. So why didn't I just turn the table around, transpose the table to put the molecules into the PC space? And the answer is quite simple. You're looking at about 12,000 molecules here right now, which will fit fine into the Excel if I transpose the table. However, the original table we were working with was about 80,000 molecules. So if I have a huge table, sometimes even performing PC will be hard. So another trick that you can remember is that even if I have PC coordinates for my descriptors, I can put the molecules in this PC coordinate space. In our platform, we will do this automatically. Let me know if you're following, because it's, again, I'm always concerned whether I'm clear on what I'm saying or not. Everybody is okay so far? Okay, great.
Yes, yes, I am.
Great. So let us, if you have that table, the original table, which is called, what is it called? It is called Toxic Metabo Descriptors 12,000 Molecules. You got the link for it. Hopefully you have the time to download. And we will upload it into utilities. And so areas of analysis, utilities section. And in the utilities section, we have this button called PCA Transform. And this button will perform all the complicated matrices and multiplication of matrices, and will also clean out the coefficients of descriptors that we don't want, the tails that I was talking about, if you remember. So we will upload the table, the file, 12,000 molecules file. And it takes some time because it is a heavy table. Cancel no groups. We will start the pipeline and we will push this PCA transform. Now there are some parameters and I want to go through them one by one and discuss them a little bit. This parameter of filtering, as we also have in PCA, for our table is irrelevant. So let's say we don't want any filtering. Is it already in natural logarithms or logarithms? Our table originally is not, but we don't want any kind of normalization. We're not interested in that. So we will put yes here so that the algorithm will not transform it into a linear, into natural logarithmic scale. So standardized table columns before PCA. What does this do? This standardizes the columns of the PCA table, of the input table. Why do we want to standardize the columns before the PCA? Well, we want to standardize the columns, and we do recommend this is by default, it's true, and we do recommend to do this, and this is to center the PCA and to make sure that you don't have any outliers that take up all the variance. So, for example, imagine that you have some graph, a lot of points are very close together, tightly clustered, and one point is very far away. So the variance of all the data set will probably fall mostly on this outlier. To reduce this great effect that this outlier will have on our PCA, because PCA coordinates are trying to maximize the variance, the explanatory variance, we will standardize the table so that our values are standard. So this will be true. Filtering PCA result table. This is what I was talking about when I said that we want to filter out these ends. We want to make sure that we're taking only descriptors who have big influence on the PC, that have big coefficients. And all the coefficients that are very small, more than 2 standard deviations from our mean, from the average, from the average coefficient, we want to get rid of them. We want to get rid of them in this specific project or this specific type of problem, so that we can afterwards make the number of these descriptors smaller. That's why we're doing this, okay? So here we will put, afterwards you can play around with the numbers, of course, but usually it's 2 SDs. When you think of the ends, you usually think of the threshold of 2 SDs. Maybe you can try with 1.5 SDs, but not less than that. SVD3 is another, is just a different algorithm of PCA. So you can use SVD2, the one that we are going to use, or SVD3. It doesn't really have a great impact. Now, the last parameter is standardizing the rows before the PCA. We also recommend doing that, even though it does change the matrix, the original data quite a lot. And again, this just means that you're making your PCA centered, and you want to have your PCA centered and not very far away. So those are the parameters. And what we will get in after we perform this will take a while, and it will. This algorithm takes a while, and it takes a while because what you're trying to do there is the following. You are taking your original table. Let me maybe demonstrate it on some Excel file. I hope it starts running in the end. Is everybody's pipeline okay? Is it just mine or? Okay, let me try again. So I upload the file, the original 12,000 molecules file. Okay. No filter. Is it on logarithmic scale? Yes. Do standardize the columns. Filter out the ends. Use SVG3, no. And standardize the rows, yes. Let's try again. Okay, so let me share with you a little bit why it takes so long. Because it does take about 7 to 10 minutes. It depends, of course, also on the amount of people doing this at the same time. So what we're going to do is, I will do it on a very small, sorry, on a very small piece of paper. Let us say that we have this table, and let us say that we have a PCA. And it doesn't really matter what I will put here right now, but we will be doing, the algorithm does the following. So I'm just writing PC1, and let's assign some random numbers to this. Doesn't matter. PC2, and again, we can just copy this. Let's say we have only 2 PCs. This is my table after I have cleaned out. Let's make some of these zeros. Those are the ones that I cleaned out, the descriptors that I thought do not have any, that have the coefficients that are smaller, that are farther away than 2 SDs from the average coefficient. So I'm going to calculate. Now, what I have here is, how do these descriptors, how do they go into my PC coordinates? So if I wanted to make a graph, I would know that on this graph, I have my descriptors. But what I actually want are my original molecules, not the descriptors. How can I project the molecules into this space? This is PC1, PC2 space. Well, I will multiply these 2 matrices. I will multiply this matrix by this one. And how do we multiply matrices? We do sum product of the matrices. So, for example, let me make it actually even smaller, just for. So here I will do my multiplication. So I take my PC table and I turn it around. Now I want this molecule to be in PC1 space. So what I will do is, let me just color this red. So I will have molecules now. In this PC1, PC2 space. What should I do with this? Equals sum of this vector and this vector. Yes, why not? Because I actually made a smaller table, forgetting that I have to have the same amount of descriptors. So of course I need just 4. 1, 2, 3, 4. The matrices have to be the same size, otherwise I can't do that. Now this molecule will also be in PC1 space. So I will do sum product of my second molecule on PC1 space, and so on. So when you have a table of 12,000 molecules on 140 descriptors, this takes about 10 minutes. Let's see if I use these 10 minutes up and we have our results. Yes, we do. Everybody, is everybody following? Are we good? Yes? No?
Yes, we are. I don't know if you see it, but in the chat, a lot of us are replying.
Yeah, if I open the chat, then then you will all see my chat, of course.
That's true. Yeah, but I think we are.
Okay, so if we're good, let me just continue. So
sorry, I have, can you show us one more time how to do the sum product?
The sum product, yeah, this is just how you multiply the matrices, and what is important is that you have 2 matrices, and the number of columns of one matrix has to be the same as the number of columns in the other. And what you do is in Excel, if you want to do it in Excel, you can write equals sum product, the first array or the first vector, times the second vector. Now the second molecule, we want it again in the PC1 space. We don't want it, if we'll take the second vector, it will be already in PC2. So all of the molecules, when I want them in PC1, I want the coefficients for PC1, I multiply the next vector by, again, PC1, and then the next by, again, PC1. And when I come to PC2, I want my first molecule in the PC2 space. I equals sum product of the first molecule in the first, sorry, not in the first, in the second.
Thank you so much.
Yeah, it makes sense. Good. Okay, great. So let's come back to our pipeline. We have three tables, three files that we will all download. And let me explain what the files are. The files are like these. You have the regular PCA result, which is unchanged PCA result as it should be. Our descriptors in PC space. Like, as you remember, we said that if we make PCA of this table, we will get descriptors in PC space. So this is what we get in this PCA result. Then we get this result corrected. Corrected, it means with corrected coefficients, so that when the coefficient is in these extremes, very small or very big, we don't want it. And the last table is the PCA transform, which is finally, after this multiplication of matrices, this is molecules in the PCA space. So this is the table we will be working with when we will be doing supervised analysis. But we should all download all of them. Why? Because we will use the other tables for our interpretation. To come back again to the descriptors from the PCA space back to the actual descriptors, we will need these tables. So save. I have it all, but let me save it again. Okay. Now, let us open this transform table. Let us open this transform table to see what's inside and to work a little bit with it to prepare the train and test tables that we need for the supervised analysis. So by doing this, we have actually gotten rid of this big problem of non-normality and dependence. And if you don't think that this was needed or this was necessary, I urge you to work with the other table and try to do the supervised analysis with the original table. Of course, you will not get very far. Actually, you will not get any results every time. And it will always tell you that you have dependencies in your table. So when you have this kind of a problem, sometimes it's not as obvious as in our table when we had ones and minus ones. Sometimes the tables look fine to us, but something doesn't let us make the supervised analysis. So when you do face these troubles, this is one of the solutions that you can use. This is actually something that people use quite often. So here are our PCs, and this name actually means, because of Unix having some special characters, we cannot put dots in the strings, in the headings, right? So we needed to make dashes or underscore. So this is actually means that this PC1 explains 7.57% of variance. and so on. PC2 explains 1.15%, and so on. Now, there is a bit of a problem starting from this CD, where you get zeros. So the easiest way is just to erase it. Now, but I think, let me just make this so that I understand how many rows I have. Let's leave only 60 PCs. 60 PCs is a lot. We will lose a little bit of information, but just a tiny bit. So let's go to about 60. It doesn't really matter where, but 60 sounds good to me. And just erase this part of the table. We are trying to make train and test tables. So here we need a word class. We will also remember that this identification T3D means that the molecule came from toxic molecules database. So we will do filtering because we need classes here. 1 will be for toxic molecules and 2 will be for non-toxic or metabolites. So here we will text search and it will contain T3D, for example. Then I know that all of the ones that I just filtered out can be made into class 1. Now, if I just select the number 1, I will be left with all the second class, the HMGB class, which is metabolites. So this here I will do 2. Just to make sure, for some reason, I still have some of these HMBDs. So let me. And now I have only 2 classes. Great, clear. So now I just made a training set for the supervised analysis, and I will save it as here. I will write the folder will be input supervised, and I will save it as, again, here you can find that in the name you have a period. Usually you don't want to risk it. So if you have a period in the name, some programs, some scripts in R don't recognize it, some Python scripts don't recognize it. So it's always better to get rid of any periods. So you can or use this dash or nothing. And I will just write that this is molecules in the PCA corrected space. And this is a train table. Now I can erase the first column. And this will be my test. Save as. Again, it's molecules, PCA, corrected space, and test. If you were not able to follow, you have the test and train set also in the links that I tried to give you. And now let us go to our supervised analysis. Hopefully you're very familiar with it. And we will run the random forest many times to identify, but again, we are trying to make the descriptors a very small number of descriptors. So we need a very small number of PCAs, because PCAs themselves are explained by a lot of descriptors. So we will run the following with the following parameters. Let me just show you the parameters that I was trying to prepare for you, so that they will, on the first side, they will separate the data quite clearly, and on the other side, they will also contain very small number of PCs for afterwards, for us to understand what it actually means that we found these PCs. How can we go back to our words, to our descriptors that will help us to take a molecule, any molecule that has been described in the database, and we will be able to identify whether this molecule is a toxic molecule or non-toxic molecule. And I'm sure you understand why it's important to understand which molecule is toxic or non-toxic. Okay, so what I chose is that the number of trees will be 50, and the number of iterations will be 10, and the number of best features, which are PCs, will be 3. So let us make this the analysis with these parameters. So we'll upload the tables, test and train tables that we just prepared. Okay. And we put them in the right groups, so that the train group will contain the train table and the test group will contain the test table. And just a little reminder, as you might remember, random forest does not have a problem over learning. However, some other discriminant analysis functions do have. For example, LDA, QDA as well, SVM, naive Bayes discriminants. All of these do have overlearning problems. So what people do is they take not a whole set as a test set, as a training set. They take a smaller set as a training set, but it has to be big enough for the model to be good. And a testing set that contains some of the samples that have not been included in the training set. So, but again, random forest doesn't have that problem, so we can, so that's why we are using the same tables for training and for testing set. And it doesn't have this problem because it is iterative, so it does the same, it chooses the features many, many times randomly. And because it also has a lot of decision trees. So random forest has 2 types of iterations inside of it. So it's a lot like bootstrapping. Okay, random forest multi-run. And here we said, just because I tested it and it doesn't take too long, and it does explain the data quite well, that's why we're using 50. And of course, I encourage you, when you do the same analysis on your own, trying to play around with the table, try something different. And you will see how the model reacts. does it predict now worse if I take a very small number of trees, or what happens to my model when I change these parameters? And also, of course, what happens to the running time? For example, in real analysis, we wouldn't use 10 iterations. We would use more like 100 iterations. But this will take a long time, especially with large tables. So a number of best features, as you remember, when we were working with genes, 50 was fine for us. 50 genes, we can easily put it into David. And I hope that David did work for you after our Friday session. Sorry. So when we were working with genes, 50 genes was small enough for us. It was good. But here, if we will choose 50 PCs, afterwards going back and finding which descriptors make contribution to the 50 PCs will be very, very hard. So we want to make this number as small as possible. And I found that 3 works okay. And frequency. We want to choose them on the amount of times we chose them as discriminants. And then we will perform LDA more for a visualization and then to try to interpret the results using the function, the LDA function, and okay. And we will run the pipeline. So now this, again, this will not be done in a second. It will take some time. So if you have any questions or comments, this is a great time while we wait for the results. I haven't been so clear in a long time. I'm very happy about it, but is this true? Okay, no questions, that's fine. Maybe I can look, maybe there is a chat that I'm not following. Okay. So if you do have questions, I will be happy to answer while we wait for our pipeline, because it will take about 10 minutes.
I have a question with the PC transform. So is it the platform automatically set it to transform the PC in linear scale after if we don't choose that LN to no?
Sorry, linear scale, what does that?
So when we are doing the parameter for the PC transform, I remember we set the LN to yes, right?
Oh, that's right.
Go ahead.
Yeah, you wanted to ask about that? Why did we do that?
Is it because the system set it to, like, the default setting is transforming the whole data set to linear scale after we do the pipeline?
Okay, so actually, maybe the problem is with the way we are identifying, naming this parameter. Let me share it again with you, and maybe it will be a little bit more clear. It asks you, and it has the little I sign. Let me just, I'm just putting in some files so that I can show you the actual parameters. So this PC transform, and it also has the same with PCA, with a regular PCA. It has this LN. And here, there is a little explanation if you just press, not press, but stand on this. What we actually mean here is that we're asking if the data that you're uploaded, that the input data, the input table that you have, is it already in natural logarithmic scale, in any logarithmic scale, natural or base 2 or base 10, whatever. Mm-hmm. And why are we asking this? We are asking this for two reasons. First is that we really suggest that you do transform your data, and it's kind of a reminder to transform your data to logarithmic scale, because logarithmic scale is much nicer distributed. So, for example, imagine you have numbers, you have expression data, gene expression data, and you have numbers such as you have expressions of one gene that is 20,000, 30,000, 25,000 across the samples. And then you have another gene that its expression is 20, 10, 15. Of course, if it's not in logs, the numbers will be, the variance of this data set will be so big. So when you will do the PCA, just accounting for all the variance, it will take a lot. But if you transform it to logarithmic scale, the numbers will be much closer together. So the ones that were in thousands, the gene that was in thousands, sorry, it will be in logarithm 9, 10, something like that. And the one that was in 20s will be logarithm 3. So the difference will not be so big. And the data will not be what we call skewed. It won't be so skewed. And it will be much better distributed. So also the PCA will look much better. Mm-hmm. But in general, of course, this is a type of normalization technique that is that has proven to be very beneficial, especially in omics, in expression analysis, in transcriptomics analysis. So this is why we're asking this. Why did we put here yes? Because our data was not in logarithmic scale. But we put here yes so that the algorithm will not transform it to logarithms, to the logarithmic scale. We put here yes to ignore, to leave our table as it is. So that was the rationale. I hope it makes sense.
Yes. So will the platform treat it as log scale instead of like
So the algorithm doesn't know what I mean. It just wants, it's just a reminder. When it sees the numbers, it just sees the numbers. It doesn't care if it's in logs or lans or any other scale. It just treats the numbers as they are. If I put here yes, it will treat the numbers as they are. If I put here no, it will transform it to logarithms. My input table, instead of being -1, it will take logarithm of -1, and so on. And of course, taking a logarithm of a negative number will be a problem.
Thank you.
No problem. Okay, maybe we can see if we are good there. Yes. Okay, great. So we got the results. Hopefully it worked for everybody. We will save the results as usual. The not the R scripts, but the zip files. So let me make a new folder and we will call it supervised analysis. The LDA zip file. And we will extract these. Okay, so as usual, we got this file that is called presents. I guess it's from the word present or presents, like gifts. So these gifts, if we open this file, it has the PCs and how many times they have been chosen. And remember, we chose only 3. So 2 of them were chosen 10 times out of 10 because that was our initial parameter that we want to do 10 iterations. So on all iterations, we chose PC2 and PC3. And on almost all, we chose PC10. This was the gift file. We don't want to save it. What else do we have in our random forest? We have this train and test tables that are only with the 3 features that we have chosen. So if we open it, we chose these 3 features, PC2, PC3, and PC10. And this is their, in genes we had expression, but here we have PCs. Now, let us now see our LDA to visualize how do these 3 PCs separate the toxic and non-toxic molecules. So we will open the LDA plot. And as I said, we chose a quite small number of PCs. However, and I'm reminding you that group 1 is the toxic molecules, and group 2 is non-toxic or metabolites. As you can see here, that they overlap. So if my LD loading has this value close to 2, it will be hard for me to say which type of a molecule it is, whether it's toxic or non-toxic. However, if my LD loading will have a negative number or even less than 2, I will almost certainly know that this is a toxic molecule, which is great. That if I get a negative loading, I know that I shouldn't be using this molecule for treating people. It can be toxic. If my LDA is here loading is close to 2 or 2, I should be very, very cautious because it might be non-toxic, but it does have some probability of it being toxic still. So this is about PCs though. This is not about the descriptors. We right now don't know which descriptors these are. But we do know that it is PC2 and PC3 and PC10. So let me just write that down, PC2, PC3, and PC10. First of all, let's check which descriptors have big contributions to these PCs and which and thus they play a role in this function, discriminant function that we found. So, as you remember, we have downloaded this PCA result corrected, the corrected PCA table that without the tails of the coefficients. So let us open this and see what we can find. Amazingly, most of them are zeros, and this exactly was our goal, to make the coefficients that are in the ends zeros, as if not making any contribution. And we remember that we are talking about PC2, PC3, and PC10 as discriminating between the toxic and non-toxic. So if I take only these three PCs, ignoring all the rest, and I will find only descriptors that have non-zero coefficients. So organochloride, metabolite, aromatic hydrocarbon, and so on. There aren't a lot. Nitrite, cephalosporin, sclerosing solution, amine, ether, antiemetic and pesticide, pollutant, and finally, lacrimator. Now, because I was doing this before, I also Googled these up to see what they mean. And most of them, except for maybe, metabolite, which is obvious, and pesticide, pollutant, most of them are chemical properties of a molecule. And I made a table of what they actually mean. So let me open it up. And show you. So these are exactly what we just highlighted together, And here you can see I made a column with description of what they are, or how they relate to toxicity. So the first organochloride is known to be toxic. Metabolite, of course, is necessary for metabolism. Aromatic hydrocarbon is known as human carcinogen. Nitrite is known poison, and so on. So how can we go back and interpret our results on the LDA table, on the LDA, how can we interpret these results? How do we identify the actual molecule, whether it's toxic or non-toxic? So to identify the actual molecule, whether it's a new molecule from the database, whether it's toxic or non-toxic, I will need to do the following. So I will take some molecule from my list, doesn't matter which one, and I will find its coefficient in the original table for these descriptors that play a role in the PC2, PC3, and PC10. Okay? So here I have a table with some highlighted columns, which actually are the descriptors that we just found with you together that make contribution to PC2 or PC3 or PC10. And here I wrote some molecule that we right now don't know what kind of a molecule it is. Then I took the PCA table for only those PCs and I transposed it. So how does this molecule look like in these PC coordinates? Well, if I take each value for each descriptor of the molecule and multiply it by the PC coefficient, I will get this molecule in this PC, on this PC vector. So I multiplied -1 times -0.26, and I got 0.26. And I did this for all of the descriptors. And I got this molecule in this 3-dimensional space. As you remember, in the LDA analysis, we also got the discriminant function. Actually, how did we get this? This is from the discriminant function. And if we open this discriminant function, we will see the loadings on the LDA1 axis. So the loadings on this axis, this is LD1 and this is LD1. I just copied this function right here. Those are the loadings of each PC. Now I want to understand how this molecule looks like on this graph. Where is it on the LDA, on the LD1? So I take the loading of PC2 and I multiply this loading by the sum of these, plus this loading by the sum of these, plus this loading by the sum of these. And I get one number, this number, -0.5. And this number tells me where this molecule is here. If it's -0.5, it probably is somewhere, I don't know, here. And because it's in the negative part of my graph, I know that this molecule is toxic. And indeed, this molecule was taken from the toxic database, from the database of toxic molecules. So it seems like at least for this molecule, our model is working fine. So in the end, what we got to is 12 descriptors and 3 PCs, based on which we can determine whether the molecule is toxic or non-toxic. please let me know if this explanation of going back from the PCs, going back to the descriptors and understanding if I take a new molecule, what happens to it? How can I see it on the graph? Where is it on this graph? If this is clear or maybe not clear enough, let me know. I know this is a lot of working with tables and a lot of working with Excel files, and sometimes it can be confusing. So let me know if you're still following. And if there are any questions, actually now is the great time, because from my side, I'm finished. If you understood everything I was talking about, and if you can do this on your own, play around with this data set, try to find a different molecule, try to understand whether we can identify it as a toxic or non-toxic by these 12 descriptors. If you could do this on your own, I think you're all set.
Okay, so if no one else has any questions, we were waiting for this lecture to send out the article review questions. So now that everybody understands the data, what it looks like, what would be the results from the analysis, we can have that sent out to everybody if everybody feels like they understand this. So please ask your questions now if there are also specific things that you might want to find out about the original data set or something like that. Otherwise, I'll send out the questionnaire today. Okay. All right. Well, thank you, Julia. This was very good. And we'll post the recording. As you all know, we now put these on YouTube, but we'll also put them on the